I have the pleasure of introducing this morning's first speaker, first abstract entitled Developmental Programming for Allergic Disease, a Secondary Analysis of the Mother's Omega-3 and Mental Health Study, presented by Dr. Vivian Romero from the University of Michigan. Good morning, Dr. Romero.
Good morning, Dr. Dalton, Dr. Garidi, members and guests. I would like to thank the Society for the opportunity to present our work. Allergic diseases affect 60 million individuals in the United States alone. Not working. Also, this one. Oh, you're right. The cost to the country is $14.5 billion per year. Allergic diseases have no cure. Treatment is symptomatic, and some preventive strategies may work. They are the fifth leading cause of chronic disease in the United States. Finally, secular studies report that the frequency has increased dramatically in the last year. In terms of burden of disease, 5 million are affected with Parkinson's, 5 million are affected with Alzheimer's, 6 with a stroke, 12 with cancer, 13 with coronary heart disease, 26 million with diabetes, and 60 million with asthma and allergies. Therefore, 1 out of every 5 Americans is affected by these disorders. Genetics play an important role in the development of allergic disorders. If one parent is affected, 1 out of 3 children will have an allergic disease. While if both parents have an allergy, 7 out of 10 children will have an allergic disease. An environmental component is essential because, by definition, allergy is an immune hyperresponse to a foreign protein or allergen. In general, immune responses have been classified into 2 types. Th1, which is implicated in protection against viruses, intracellular pathogens, and neoplastic cells, while Th2 immune response has been implicated in protection against parasites, maternal tolerance of fetal antigens during pregnancy, and in the genesis of allergic disorders. Th1 immune responses are mediated by specific cytokines and chemokines, such as interleukin-1, interferon gamma, and the chemokine interferon gamma-inducible protein 10, or IP10. In contrast, Th2 immune responses are mediated by interleukin-4, interleukin-13, macrophage-derived chemokine, or MDC, and thymus-activated chemokine, regulated chemokine, or TARC. In the presence of interleukin-4 and interleukin-13, B lymphocytes are generated, which in turn can produce immunoglobulin E, which are the antibodies in allergic disorders. A fundamental question in the understanding of allergy is whether it can begin during fetal life, since children are frequently affected. Several lines of evidence support that this might be the case. The human fetus is exposed to allergens. This is a house dust mite, which live in the dust of all our homes. In the year 2000, investigators reported that the antigens of these dust mites could be detected in amniotic fluid and umbilical cord in normal pregnancy, and this indicates that the human fetus can be exposed to allergens. Once the requirement of environmental exposure is met, the development of allergies require a Th2 bias immune response. How can I go back? Okay. Under normal circumstances, pregnancy is a state in which there is a Th2 bias immune response, and this may favor the development of an allergic response. Recent observations indicate that fetuses with an excessive Th2 immune response, determined by the concentrations of chemokines in umbilical cord, are at increased risk of developing childhood allergies. Evidence supports that the ratio of macrophage-derived chemokine, or MDC, to IP10 is higher in the umbilical cord of children who subsequently develop allergic symptoms in comparison with those that did not. Obstetrical factors that may predispose to allergic disorders in the offspring include cesarean deliveries. At least 2 meta-analyses have provided evidence that allergic disorders are more frequent in infants born after cesarean section. Other factors include nutrition during pregnancy. The Western diet, for example, is rich in omega-6 fatty acids. In contrast, the Mediterranean diet is rich in omega-3 fatty acids, which has been implicated in the protection against a Th2 immune response and therefore allergies. Our hypothesis is that fish oil supplementation during pregnancy can affect the ratio of Th2 to Th1 chemokines in fetal life, as reflected in umbilical cord blood at birth, and consistent with a profile reported to be protective against allergic disease in the offspring. The objectives of this study were, first, to determine if umbilical cord and maternal plasma concentrations of chemokines associated with the subsequent development of allergic disease in children change after maternal supplementation with fish oil of eicosapentaenoic acid, or EPA from now on, or docosahexaenoic acid, or DHA, versus placebo. Second, to examine if the mode of delivery, vaginal versus cesarean, is associated with changes in the chemokine profile associated with the subsequent development of allergy in children. This was a secondary analysis of the mother's omega-3 and mental health study, a randomized clinical trial performed at the University of Michigan. The design, analysis, and results of this study were presented yesterday as part of abstract number 29 in the oral concurrent session. In this trial, mothers were randomly allocated to receive placebo, EPA, or DHA. Maternal and umbilical cord blood were obtained during pregnancy and at the time of delivery. Chemokine analysis was performed using immunoassays using commercially available methods. Descriptive statistics were computed according to treatment arm, and log transformation was performed for variables not following a normal distribution. For comparisons of continuous variables between groups within each study visit, ANOVA or 2-sample t-tests for independent samples were used. For comparison of categorical variables, Fisher exact test was used. Multiple linear regression was used to examine the association between treatment arm and chemokine levels while adjusting for covariates including gestational age, birth weight, and mode of delivery. A p-value less than 0.05 was considered statistically significant. Results. There were no differences in the clinical characteristics among the 3 groups: EPA-rich fish oil, DHA-rich fish oil, and placebo. There were no differences detected in the unadjusted mean maternal plasma chemokine ratios, MDC to IP10, or UNTARC to IP10 among the EPA, DHA, and placebo group at each visit. The mean chemokine ratios and umbilical cord plasma were lower in women who received EPA or DHA than those who received placebo. However, this did not achieve a statistical significance. Adjusting for mode of delivery, gestational age of delivery, and birth weight, the ratio of log MDC to log IP10 was 1.8 units lower, right here, in the EPA fish oil group, and 2.0 units lower in the DHA enriched fish oil compared to placebo. And this was statistically significant. The ratio of log MDC to log IP10 was 1.6 units higher in women undergoing cesarean section compared to those undergoing vaginal delivery, and this was statistically significant. There was no significant effect of gestational age of delivery and birth weight on the ratio of log MDC to log IP10. Similar results were obtained for the ratio of log TARC to log IP10. Conclusion. The principal finding of this study was that prenatal supplementation with EPA and DHA-rich fish oil is associated with a change in the chemokine profile away from a predominant Th2 response. Second, cesarean delivery was associated with a change in the chemokine ratio favoring a Th2 response. Our results may provide a possible mechanisms to explain findings from prior epidemiologic studies showing that maternal diet and mode of delivery may increase the risk of allergic disease in childhood. With gratitude to my co-investigators at the University of Michigan, and in particular to Dr. Mazurkiewicz for her mentorship. I would like to acknowledge all the people that collaborated in the performance of this study. Thank you, and I'll be happy to take any questions.
Paper is now open for questions.
Hey, Tom. Sabai from Houston. Very nice presentation. I didn't see about your cesarean sections. Were these elective, or these are women who had labor? And how are these will affect your level? And second, I'm not clear, did you measure the concentration umbilical vein or umbilical artery? And do you expect to find differences between the two?
Thank you, Dr. Sabai, for the question. For the first one, actually, we were planning to conduct a sub-analysis where we were going to look at the difference between the chemokine ratios in the patients who had planned cesarean section versus patients who presented in a spontaneous labor and have failure to progress or any other reason and end up with a cesarean section. We do have analyzed that data, but unfortunately I don't have the results to be able to share with you at this time.
How about the cord? What did you measure? Was it in the artery or the vein?
We actually, we didn't, we just collected cord blood at the time of delivery, and typically it's umbilical vein. I don't see there are any studies that actually have reported if there are any difference between the chemokine ratios, if it's either in the umbilical cord, vein, or artery. I'm not aware of that, but thank you for the suggestion.
Other questions? Can you speculate for me, and I may have missed it, I apologize, but how you might see, I can understand how the diet might alter the ratios, but how might the process of labor or cesarean section actually affect things?
Can I have, let me see.
I'm not the straight man here to bring up her extra slides. So.
Thank you. So there's something called the hygiene hypothesis, and there is a theory that the children born by cesarean section has been shown to have a delay and altered development in the establishment of the gut flora, and for that also alter cytokine and probably chemokine production. There is something about the kids that, the children that are delivered by C-section, they're not exposed to the vaginal flora, and for that reason, there are studies actually that have already showed that there are differences in the microbiome in the gut of children that were delivered by vaginal, by cesarean section versus vaginal delivery.
Thank you. Dr. Spahn?
Lovely presentation. Thank you so much. Can you give me what you would tell a patient, given what you've seen on the changes of the TH1 versus the TH2, using DHA or the omega-3? Would you recommend that they take them or not take them?
Thank you for the question. That's a very interesting question. Well, we have to remember that this is just a secondary analysis of a randomized controlled trial. This study wasn't designed to look at that particular question. We actually, with this study, what we were looking was just to see what was the effect of fish oil supplementation on the chemokine ratios. I think that in order to be able to answer what the question, we'll have to design like a larger trial, randomized controlled trial, longitudinal study that actually looked not only at the effect of fish oil supplementation, but also taking into account like the genetics, the environment, and also DNA variants, and so that we will be able to, come with a stronger data and say that, yeah, that fish oil may be helpful for prevention of allergy. Thank you.
Thank you very much.
Thank you so much for those kind words, and thank you, Drs. Garidi and Dalton. And we'd also like to thank the Society for the opportunity to present our research here today. It's a true honor. Maternal obesity is a transgenerational health problem. 1 in 3 women in the United States is now obese at the time of conception, and there's mounting evidence of neurological morbidity in the offspring of obese women. Compelling data from large epidemiologic studies have demonstrated a link between maternal obesity and adverse neurodevelopmental outcomes in offspring, including lower cognitive performance, an increased incidence of autism spectrum disorders and developmental delay, and an increased incidence of ADHD. Rodent models have been utilized to explore the possible mechanisms by which maternal obesity might impart an increased risk for adverse neurodevelopmental outcomes in offspring. These models have highlighted both structural brain changes and differences in brain gene expression in the offspring of obese female rodents. The structural brain changes noted have included differences in brain apoptosis, as well as differences in the neuronal migration of stem-like cells in the fetal brain. While we commonly perceive apoptosis to be a response to harmful stimuli, apoptosis actually plays a critical role in normal neurodevelopment. On the right, you can see a figure from a seminal paper demonstrating fetal brain development in wild-type mice compared to brain development in mice who are deficient in Casp9, which is a gene whose protein product is critical to the final common apoptotic pathway. So you can see here that on embryonic day 10.5 at the top, there's increased survival of neural progenitor cells in the fetal brain, ultimately leading to exencephaly, a severe brain anomaly, by embryonic day 16.5. So we know that apoptosis plays a critical role in normal neurodevelopment and that there are differences in apoptosis in the brains of offspring of obese female mice. Prior work by our lab has demonstrated that cell-free fetal RNA in amniotic fluid provides valuable real-time fetal gene expression data and therefore may be a window into fetal brain development during this time period. To our knowledge, no studies have yet performed global gene expression profiling in fetuses of obese human women. We therefore sought to perform a functional transcriptomic analysis of amniotic fluid cell-free fetal RNA in obese compared to lean women in the second trimester. We wanted to determine which genes are differentially expressed in fetuses of obese women to elucidate the possible molecular mechanisms underlying an increased risk of morbidity, specifically neurodevelopmental morbidity, noted in offspring. Yesterday in abstract number 4, we presented a separate set of findings from this same study design, highlighting a possible fetal origin of metabolic syndrome. Today, utilizing the same study design, we'll present these distinct findings pertaining to fetal gene expression as it might be involved in neurodevelopment. This was a prospective study, including women undergoing genetic amniocentesis in the second trimester for standard clinical indications. The study population included 8 obese women with a BMI greater than or equal to 30 and 8 lean controls with a BMI less than 25. All fetuses were singletons and had normal karyotype, and we excluded fetuses with structural anomalies. Obese cases and lean controls were matched for gestational age and fetal sex, both of which have been demonstrated to influence gene expression. mRNA was extracted from amniotic fluid supernatants, converted to cDNA and amplified using commercial kits, fragmented, labeled with biotin, and hybridized to Affymetrix whole genome arrays for gene expression analysis. We identified differentially regulated genes in fetuses of obese women compared to lean using the dependent t-test with a Benjamini-Hochberg correction to limit the false discovery rate, given the multiple comparisons being made. To give you a sense of how we arrived at the final gene list, the Affymetrix array that we use contains more than 54,000 probe IDs, or oligos, that interrogate more than 38,500 genes. We consider genes to be significantly up or down regulated in fetuses of obese women if they were up or down regulated in 8 out of 8 pairs and associated with a significant corrected p-value of less than 0.05. These genes formed the final list that we uploaded to Ingenuity Pathway Analysis, or IPA, for a functional transcriptomic analysis. IPA is a publicly available bioinformatics database that's able to identify specific biological processes, molecular and cellular functions, and dysregulated physiological systems and signaling pathways within your data set. We use standard significance cutoffs for this type of microarray work. You can see in our results table that the obese and lean study populations did not vary significantly with respect to any of the demographic variables apart from maternal BMI. We identified 205 differentially regulated genes in fetuses of obese women compared to lean. 114 of these were significantly upregulated, while 91 were significantly downregulated. What emerged was a picture of decreased apoptosis in fetuses of obese women. We found that genes involved in resistance to apoptotic cell death were significantly upregulated in fetuses of obese women, while pro-apoptotic genes were significantly downregulated. This graphic is to highlight the role of specific genes that were significantly dysregulated in fetuses of obese women and to demonstrate how they decrease apoptosis. So this is the intrinsic or mitochondrial pathway of apoptosis, and it's initiated by either toxic insults or normal developmental cues. Pro-apoptotic molecules will then induce the release of cytochrome C from mitochondria, ultimately activating the final common apoptotic pathway. This could be disrupted in several different locations. In fetuses of obese women, BCL2 and BCL2L1 were both significantly upregulated, therefore blocking the activation of mitochondria by these pro-apoptotic molecules and decreasing apoptosis. Cas9, whose protein product is critical to the final common apoptotic pathway, was significantly downregulated in fetuses of obese women, so this also decreased apoptosis in these fetuses. So pro-apoptotic genes are significantly downregulated in fetuses of obese women, and anti-apoptotic genes are significantly upregulated. But you may be wondering, what does this really have to do with the central nervous system? And this is where our functional analysis on IPA really came into play. We found that apoptotic cell death was significantly downregulated in fetuses of obese women throughout, but that this was particularly striking in pathways pertaining to the central nervous system. The most significantly dysregulated molecular function in fetuses of obese women was cell death, with significantly decreased apoptosis of cerebral cortex cells being noted, as well as decreased cell death of hippocampal cells. And we also found that apoptosis of sympathetic neurons and cell viability of dendritic cells were significantly affected in fetuses of obese women. These were the genes that Ingenuity Pathway identified as being implicated in dysregulated central nervous system apoptosis in fetuses of obese women. You can see that regardless of the direction of expression in fetuses of obese women, with upregulated genes being on the top in the lighter color and downregulated genes in fetuses of obese women being below in the dark blue, that the direction of expression in these fetuses all favored decreased apoptosis. So using the oligos or probe IDs on the Affymetrix array that interrogated these specific genes that were identified by IPA as being implicated in CNS apoptosis, we performed a principal component analysis. A principal component analysis utilizes a computer algorithm that's blinded to phenotype. So the computer has no idea that these women are obese and lean or on what basis they vary. We upload our gene expression to the computer program and ask it to group the data based on variables, also called components, that are accounting for the greatest proportion of differences in gene expression in our sample. So if you look here at the x-axis, which is principal component 1, and draw an imaginary line through 0, You can see that the obese women, represented by the red dots, are all to the left of 0, while the lean women, represented by the black dots, are to the right of 0. So genes involved in central nervous system apoptosis are therefore segregating on the basis of maternal BMI, suggesting that maternal BMI is principal component 1, accounting for the greatest proportion of differences in gene expression here. Finally, we wanted to mention a potential role for ApoD, which was the most upregulated gene in fetuses of obese women in fetal neurodevelopment. We know that ApoD is involved in lipid metabolism and cholesterol homeostasis, but ApoD is also highly expressed in the central nervous system, and increased expression has been noted during human brain development and maturation. The protein product of the gene has been demonstrated to exert neuroprotective effects in a rodent model of excitotoxic and traumatic brain injury, possibly by preventing against oxidative stress-induced damage. However, overexpression of ApoD during development may be deleterious, as we know from rodent models in which overexpression of ApoD has been associated with psychosis and schizophrenia. This is an image from an atlas of mouse transcriptome gene expression and depicts the expression of ApoD in the fetal nervous system. So in this sagittal section through an embryonic day 14.5 mouse, you can see that ApoD, which is staining darker purple, maps strongly to the fetal brain, spinal cord and meninges, and other central nervous system related structures, including the nose and the ear. We therefore believe that ApoD and its significant upregulation in fetuses of obese women likely plays a role in neurodevelopment in these fetuses. In conclusion, our analysis of cell-free fetal RNA in the amniotic fluid of obese women demonstrates differences in gene expression as early as the second trimester. These differences included significant downregulation of pathways implicated in brain apoptosis and significant upregulation of the gene apolipoprotein D. Decreased brain apoptosis and overexpression of ApoD therefore warrant further investigation as potential mediators of neurodevelopmental morbidity in the offspring of obese women. The molecular mechanisms underlying the increased risk for these fetuses may be initiated as early as the second trimester. And given the epidemic of maternal obesity in this country, we hope that these preliminary findings in a small group of women will form the foundation for future research and hopefully interventions. There are so many people to acknowledge, but I'd love to acknowledge Dr. Bianchi first for her outstanding mentorship, as well as Dr. Norwitz and Dr. Crago from my division at Tufts, everyone in the Bianchi lab and at the Mother Infant Research Institute where this research was conducted, and the NICHD for the funding to conduct this research. Thank you. I'd be happy to take any questions.
This paper is now open for questions.
Hi. Bakshi, New York City. Congratulations, wonderful work. The question which I have is, are these obese women excluded, did you exclude diabetes in them? And secondly, since we are talking about apolipoprotein, etc., did you have any levels in the mother, whether they had hypertriglyceridemia or hypercholesterolemia, etc.?
Thank you so much. That's an excellent question. We were not able to exclude diabetes or other pre-existing conditions from these mothers. We collected this under a discarded fluids protocol. So that is a limitation of the study, that we were unable to evaluate the maternal conditions in this case, and they certainly could influence gene expression in theory. And we also did not collect maternal blood at the same time to look at gene expression for ApoD. But in future studies, we have collected prospective data from subjects that were consented and enrolled and not using discarded materials. So we will look at ideas similar to that. Thank you.
Ram Menon, Galveston. Excellent presentation. One question and one comment or two. How much of these amniotic fluid RNA expressions are indicative of a tissue-level apoptotic changes in the fetus? And my second, comment is, in obesity, oxidative stress is one of the key factors that can also drive some pathways. And in the absence of apoptotic changes, is it possible to have an alternate pathway mediated through senescence, contributing to the pathology?
I'll answer your first question first, and I may ask you to repeat your second question. I want to make sure that people understand that we did not observe decreased apoptosis in the brains of these human fetuses, but simply gene expression that was in a direction that suggested decreased apoptosis, and using these bioinformatics tools that we unfortunately have to use to infer human biology, it was suggestive of decreased apoptosis, specifically in central nervous system pathways. So we can't demonstrate that these mRNAs are specifically mapping to the fetal brain or showing this decreased apoptosis with immunohistochemistry. And can you repeat the second question?
Now,
in essence?
why don't we ask you to discuss the second question with him after the session? Thank you.
Okay.
Thank you very much.
Thank you very much.
Super job.
Thank you very much. I would first like to acknowledge my co-authors, Dr. Dennis Ockley and Dr. Judette Lewis. Obstructive sleep apnea, or OSA, is an obesity-related comorbid condition. As obesity rates increase across the United States, there has been a growing recognition and diagnosis of obstructive sleep apnea, or OSA, among pregnant women. OSA is characterized by recurrent upper airway collapse and hypoxemia reoxygenation cycles during sleep. There is a consistent link between OSA and underlying cardiovascular disease in epidemiologic and cohort studies. This concept has biologic plausibility. Recurrent hypoxemia and reoxygenation cycles trigger a cascade of sympathetic activation, oxidative stress, inflammation, endothelial damage, and ultimately cardiovascular disease. Moreover, OSA treatment has been shown to improve cardiovascular function. However, much of the relationship between OSA and cardiovascular disease remains the subject of ongoing investigation. Our primary aim was to determine the incidence of abnormal echocardiogram findings among reproductive age women with OSA and cardiopulmonary symptoms. Our secondary aim was to compare the frequency of abnormal echocardiograms based on pregnancy status. We performed this retrospective cohort study at our academic tertiary care center. Using a sleep lab database, we identified women aged 15 to 45 who underwent a diagnostic nocturnal polysomnogram for symptoms suspicious of sleep apnea between the years 2005 to 2012. Of the women with polysomnogram diagnosed OSA, we identified women who had undergone transthoracic echocardiograms, or TTEs, within 2 years of polysomnogram for cardiopulmonary symptoms via individual chart review. We excluded echocardiogram studies that were non-diagnostic or of poor quality, in addition to women with known congenital or infectious cardiopulmonary disease, acute pulmonary embolus, or peripartum cardiomyopathy. All women underwent a diagnostic nocturnal polysomnogram. The apnea-hypopnea index, which is the number of apneas and hypopneas per hour of sleep, was calculated using American Academy of Sleep Medicine definitions. And women with apnea-hypopnea index of 5 or greater were diagnosed with OSA, which was graded as shown. Echocardiogram findings were classified according to the American Society for Echocardiography guidelines, in which mild, moderate, and severely abnormal cutoffs are defined. Findings of chamber dilation, left ventricular hypertrophy, systolic and diastolic dysfunction, valve regurgitation, and estimated pulmonary hypertension were recorded. As an example, we classified an ejection fraction of less than 55% as any systolic dysfunction, and an ejection fraction of less than 45% as moderate to severe systolic dysfunction. The primary outcome measure was the presence of any abnormal echocardiogram finding. The secondary outcome measures were the presence of a moderate to severe echocardiogram abnormality and the presence of abnormalities based on pregnancy status and comorbid conditions. As this was an exploratory study, no a priori power analysis was performed. We used the student t-test for continuous data, chi-squared and Fisher exact tests for categorical data, and the Mann-Whitney U-test for ordinal and non-normally distributed data. And now for the results. Of 1,452 women who underwent nocturnal polysomnogram, 1,265 women, or 87%, were diagnosed with OSA. Of these women, 430 underwent transthoracic echocardiogram for cardiopulmonary symptoms. Of the included echocardiograms, 92% were performed within 1 year, and 66% were performed within 6 months of OSA diagnosis. 99 echocardiograms were excluded, the majority for being greater than 2 years from polysomnogram or being a non-diagnostic study. And the remaining 331 studies were included for further analysis. We looked at the demographic characteristics of the 331 women with OSA who underwent echocardiogram for cardiopulmonary symptoms. The most striking thing we noted was the extreme obesity of the cohort, with a median body mass index of 42 kilograms per meter squared. They were older, with a median age of 39 years, they had moderate sleep apnea severity, and they were Paris. Not surprisingly, we found that the cohort had a high prevalence of comorbid conditions. There was a 60% prevalence of class 3 obesity, or a body mass index of at least 40 kilograms per meter squared. 50% of these women had moderate to severe OSA, and 46% had hypertension. 14% already carried a diagnosis of cardiovascular disease, which was defined as angina, atrial fibrillation, supraventricular tachycardia, cardiac arrhythmia, congenital heart failure, and coronary artery disease. Now for our primary outcome. We found a 51% incidence of any echocardiogram abnormality among these reproductive age women with OSA and cardiopulmonary symptoms. Based on the American Society for Echocardiography classification system, 31% of the cohort had abnormalities that were considered moderate to severe. We then examined the occurrence of individual echocardiogram findings. The blue bars are the percent of women who demonstrated the individual abnormality, and the red bars are the percent of women in whom the abnormality was moderate to severe. They are not meant as a comparison to each other, and as such, a p-value is not displayed. Of note, more than one abnormal finding was at times observed in the same woman. Chamber dilation was very frequent, as can be seen. 35% of women with OSA had any degree of cardiac chamber dilation, and at least half of those were moderate to severe. 18% of women had any, and 10% had moderate to severe left ventricular hypertrophy. 15% of women had any, and 3% had moderate to severe valve insufficiency. Systolic dysfunction, diastolic dysfunction, and pulmonary hypertension were present in the range of 6 to 10% of women, and they were moderate to severe in 3 to 5% of the total cohort. We then examined the frequency of an abnormal echocardiogram finding based on the presence of each medical comorbidity. As can be seen, there was a high frequency of echocardiogram abnormalities for all of the comorbidities examined. And, as would be expected, of the 45 patients with known cardiovascular disease, 85% had an abnormal echocardiogram. Of the remaining comorbidities, approximately half of women with class 3 obesity, hypertension, moderate to severe sleep apnea, age over 40 years, and diabetes had an abnormal echocardiogram finding. Now moving to our secondary analysis, in which we compared the echocardiogram findings by pregnancy status. Of the 331 patients with polysomnogram diagnosed sleep apnea and an echocardiogram for cardiopulmonary symptoms, 36 women, or 11%, were pregnant, and 295 were not pregnant. Of the pregnant patients, the median gestational age at time of polysomnogram was 18 weeks, with an interquartile range of 4 to 28 weeks. The median gestational age at time of echocardiogram was 23 weeks, with an interquartile range of 15 to 29 weeks. We compared demographic characteristics by pregnancy status and found that pregnant women were younger, more obese, with similar OSA severity compared to the non-pregnant cohort. When we examined the medical comorbidities of the pregnant and non-pregnant patients, we found that they were not different, except that the pregnant women were more obese. We then examined the primary outcome based on pregnancy status. We found that the presence of any echocardiogram abnormality and moderate to severe abnormalities were just as common among the pregnant and the non-pregnant women. When we looked at individual echo abnormalities, we found no differences between the pregnant and the non-pregnant patients. Chamber dilation and valve insufficiency can be found in normal pregnancies. However, other cardiac abnormalities that would be considered pathologic regardless of pregnancy status persisted among the pregnant patients as well. Specifically, pregnant women had an 18% incidence of left ventricular hypertrophy, a 7% incidence of pulmonary hypertension, and a 6% incidence of systolic dysfunction. Among the pregnant patients, we observed no difference in the frequency of a normal versus an abnormal echocardiogram based on the presence of comorbidities. These findings are, of course, limited by the small number of pregnant patients. In conclusion, this was a study of reproductive age women with polysomnogram-proven obstructive sleep apnea who underwent echocardiogram for cardiopulmonary symptoms. Half of these women had an abnormal echocardiogram finding. Our subset of pregnant women had a similarly high frequency of abnormal echocardiograms. Both pregnant and non-pregnant women had significant comorbidities that limit our ability to differentiate causality from association. To our knowledge, the cardiac abnormalities among pregnant women with OSA have not previously been described, but the high frequency that we observed is concerning. These preliminary observations provide the basis for future prospective investigations that should include both symptomatic and asymptomatic women and consider the presence or absence of medical comorbidities. I would like to sincerely thank the following of my colleagues at Case Western Reserve University Metro Health Medical Center for their significant contributions to this project, and I thank you again for the opportunity to present our findings. I'd be happy to answer any questions at this time.
Let me start with a question, please. I know that the number of pregnant women were fairly limited, and I noticed that your average gestational age was fairly early in pregnancy at 18 weeks. Were you able to segregate the earlier and later gestational ages and see whether the advancing pregnancy had any effect on their echocardiogram findings?
You know, that is a great question, Dr. Garidi, and I think that that would be a very interesting thing to look at. We were hesitant to look at that given our very small sample size of pregnant patients, but certainly were we to have a larger sample size, it would be very interesting to look at the variations by gestational age in both the polysomnogram and the echocardiogram abnormalities.
Yes, sir.
Larry Newman, Kaiser Permanente, Oakland. Dr. Vricella, that was very interesting study, and glad to see it coming. In our offices a few years ago, we had to make our scales go from 350 to 500 pounds because 350 doesn't work anymore. In your study of the 330 patients or so, do you have any information how many were being treated with CPAP, pregnant or not pregnant, and did that make a difference?
So that's a very good question, and the way that we designed our analysis was that we only chose patients who had recently undergone a diagnostic study for sleep apnea, with the understanding that previous to that they had not carried a diagnosis, therefore would not have been under treatment.
Thank you.
Dr. Coy.
Thank you. Thank you very much, Aaron Coy from Oregon Health and Science University. And I appreciate the work that both yourself and Dr. Lewis, your senior author, has been doing in this area. Incredibly important work. And there's obviously this interaction between OSA and hypertension and diabetes and obesity. And I'm wondering, what kind of other information you had on these women related to either treatment for hypertension, treatment for diabetes, or treatment for the OSA, specifically, obviously, CPAP, to look at in terms of these outcomes.
Thank you very much, Dr. Coy, for your question. Yes, as I stated, none of these women were under CPAP treatment at the time of the polysomnogram or the echo, as far as we could tell retrospectively. And as far as questions about whether they were undergoing treatment for hypertension, many of them were.
Thank you very much.
Thank you.
Thank you, Dr. Dalton. Thank you, Dr. Grady. I'd also like to thank the Society of Maternal-Fetal Medicine for letting us have this opportunity to tell you about our research, seeking to identify predictors of recurrent preterm birth using proteomics. The prevention of preterm birth is one of the most important unresolved issues confronting obstetricians, as its incidence continues to rise worldwide. Currently, it complicates 12% of U.S. births, and its incidence has increased by a third in the last 3 decades. It's responsible for 80% of neonatal mortality and the majority of neonatal morbidity. The biggest risk factor for future spontaneous recurrent preterm birth is a prior history of preterm birth. Prior studies have examined markers in the cervical vaginal fluids, amniotic fluid, and maternal serum using mass spectrometry, but findings have not been adequate for clinical prediction, and subsequent studies are ongoing. Proteomic profiling can analyze multiple protein signatures concurrently, so it's well suited for the study of complex conditions such as recurrent preterm birth. Initial studies using proteomic profiling has suggested that there may be unique protein biomarkers for recurrent preterm birth. However, many studies, including our own, have generally reported the results of pooled specimens. The findings of prior studies have led us to conduct a prospective study on a larger cohort of women. Challenges of proteomic analysis include that it's expensive and time-consuming, thus limiting individual analysis. With this in mind, our study hopes to expand on standard mass spectrometry analysis by performing ELISAs on individual specimens to determine the predictive capability for individual proteins. The objective of the study was to identify cervical vaginal fluid proteins as predictors of recurrent preterm birth in women prior to the onset of labor. We conducted a nested case control study within a very specific cohort of 107 high-risk women who were all multiparous and all prior pregnancies were preterm births. This was done in an attempt to highlight a somewhat homogeneous population that was at greatest risk for preterm birth. Subjects were excluded if they were nulliparous, had a prior term delivery, had multifetal gestation, ruptured membranes, major fetal anomaly, or chromosomal aneuploidy. After exclusions, 97 subjects remained. CVF collections were performed every 2 weeks starting at 12 weeks gestation, and this was done to identify proteins as a signal of preterm birth prior to the onset of clinical symptoms. Prior studies performed in our lab previously only looked at one time point to predict preterm birth. This study design allows us to have sample collections within 1 to 2 weeks of symptoms to better capture the protein of interest. Subjects were further stratified by term versus preterm deliveries. Of the 26 preterm subjects remaining, we had 9 medically indicated preterm births, and 17 had spontaneous preterm births. Of those 17, we identified 10 cases that had CVF samples collected within 2 to 14 days of delivery and matched them to 20 gestationally age-matched controls. Our study population was largely Caucasian and Latina, approximately 35 years old, and it had a mean parity of 2. 40% of cases used progesterone, and the average gestational age at delivery was 33.6 weeks for cases and 38.5 weeks for controls. Large-scale bottom-up proteomic methods were used. Individual CVF samples were pooled into 2 control pools and 1 recurrent preterm birth cases pool. The samples were then precipitated with acetone, digested with trypsin, and then subjected to high pH reverse phase chromatography. 80 fractions were then concatenated into 20 fractions, and fractions were then analyzed using linear ion trap mass spectrometry. Mass spec data was then analyzed using the sequest-PAUSE software and then human sprout protein database to identify 584 proteins and use spectral counting to quantify the detected proteins. Relevant statistical tests were then used to identify differentially expressed protein candidates and ELISA assays were then used to validate some of these candidates. Abundant dependent spectral count properties were then used with Z-score and chi-score tests to find differentially expressed protein candidates. Combined Z-score p-value and chi-score p-value thresholds were then used to categorize candidates by their differential expression. The blue here represents no differential expression between cases and recurrent preterm birth subjects, whereas the red, yellow, and green were all differentially expressed. Red had the lowest level of differential expression, yellow had a medium level of differential expression, and green had the highest level of differential expression, representing the proteins of greatest interest and potential biomarkers. Proteins from these three groups were then mapped to different protein pathways. We identified a total of 584 proteins. 72 were differentially expressed candidates, and of those, 38 proteins were highly differentially expressed with a p-value of less than 0.01. In addition, we identified 2 pathways that appeared biologically relevant to preterm birth: immune response modulators and extracellular matrix proteins. An overview of the 72 differentially expressed candidates were presented yesterday in Abstract 351, so I won't be presenting that data here today. Instead, I'll be focusing on the analysis of 4 targeted proteins. Of the 38 highly differentially expressed proteins, we identified 15 proteins of greatest interest based on the combination of differential expression and spectral counts. We selected these 4 here, highlighted in red, for further analysis with ELISA. In the ELISAs we're going to discuss, we matched cases and controls at a 1-to-1 ratio for analysis. These 4 proteins were selected for initial validation because they were associated with the 2 pathways of interest and also had moderately large spectral count differences. The first 2 proteins are found to be significantly upregulated, and the latter 2 significantly downregulated compared to controls. I plan to address the description and function of each of these proteins in the subsequent slides. First, CRIS3 is a matrix protein involved in immune response. Here we show a scatter plot of the ELISA results of CRIS3. The cases are demonstrated in red and the controls in blue. This will also be the case for the subsequent slides as well. The ELISA on CRIS3 demonstrates no protein expression in cases and controls except in one subject, right here, who delivered at 24 weeks gestation, where it was strongly expressed. Because of the strong expression of CRIS3 in this one subject, it's likely that this led to an overrepresentation of CRIS3 in the pooled mass spec data, which is one of the drawbacks of using pooled specimens. Next, we have heat shock protein beta 1, which is found in the cytoplasm, especially of nerve and muscle cells. It is involved in muscle contraction, cell motility, and immune response. It's also expressed in response to environmental stress. The ELISA results for heat shock protein beta 1 are demonstrated here on the graph on the left. The ELISA results demonstrate that there is a decreased expression in cases compared to controls, which is different from the mass spec results, which showed an increase in cases compared to controls. On the right, we have a scatter plot demonstrating protein expression by gestational age distribution, noting no significant difference between cases and controls. Next, psoriasin is also located in the cytoplasm and involved in immune and inflammatory response. The ELISAs for results for psoriasin are demonstrated here on the graph on the left. The results noted a similar expression between cases and controls, which is different than the results on the mass spec data, which noted a decreased expression in cases compared to controls. The scatter plot here on the right demonstrates no gestational age effect on protein expression between cases and controls. Lastly, enolase 1 is also located in the cytoplasm and involved in immune inflammatory response. The ELISA for results for enolase 1 are demonstrated in the graph on the left and show a decreased expression in cases compared to controls, which is in agreement with the mass spec results. On the right, the scatter plot demonstrates protein expression by gestational age distribution, demonstrating no significant difference between cases and controls. So in conclusion, the mass spec results identified 2 potential pathways for recurrent preterm birth: immune and inflammatory response and extracellular matrix proteins. Unfortunately, the 4 individual protein candidates we analyzed were not significantly different from controls in our initial confirmatory ELISAs. Our study has demonstrated that mass spectrometry can be applied to complex conditions such as recurrent preterm birth, as it's useful for comprehensive protein identification. But potential biomarkers need to be validated by a confirmatory test such as an ELISA or a Western blot to determine protein expression in individual subjects. The limitations of our study were that the study design has only highlighted proteins that are markers of interest, but cannot determine clinical usefulness. Our data cannot be used to answer the question of how accurate these proteins would be as predictors of recurrent preterm birth, so future prospective cohort studies are needed to validate potential biomarkers. Also, we studied a very specific high-risk population, so any findings may not be generalizable to other populations. Looking ahead at future directions, our first future plans for our study include studying the other 11 highly differentially expressed CVF proteins based on function and spectral counts, and to perform confirmatory ELISAs to determine whether they would be potential predictive markers. Second, if we identify viable predictive protein candidates, we plan to perform ELISAs on each subject's serially collected CVF samples to identify a pattern of protein expression to establish a gestational age window for optimal sampling. Lastly, further large prospective cohort studies are needed to determine predictive accuracy by looking at the number of people who have differentially expressed protein candidates and how many of them actually go on to have preterm birth. I'd like to thank the March of Dimes Prematurity Research Initiative and the Silver Foundation for the funding of this study. I would also like to thank all the people who helped make this study possible. A large and special thanks to my mentor on this project, Dr. Pereira, Dr. Reddy and Dr. Wilmarth in the Proteomics Core, Dr. Roberts and Dr. Frias for their mentorship and support, Janice and Monica for their recruitment and help and also for their support on this endeavor. And lastly, Dr. Nagala and Dr. Gravitt for their mentorship and support. Thank you very much.
Thank you very much. This paper is now open for questions. Dr. Spong, I see you going to the microphone.
Kathy Spong from Bethesda, and congratulations on a huge undertaking, and I commiserate with you on finding something and then not being able to validate it with ELISA, but great job pulling it all together and explaining it to us. I wonder, are these different across gestation, and might it be, if I understood correctly, and please correct me if I'm wrong, you used the sample that was within 2 weeks of delivery, but you had a large gestational age window. So it might be that the expression that occurs at 24 weeks is different than that occurs at 34 weeks. And so might it be that that could explain why you didn't find a difference, and maybe there is one if you were able to look at it across time?
Thank you, Dr. Spong, for your question. We are hoping to answer that question by looking at the serially collected CVF samples and performing ELISAs on that so that we can see, hopefully, a gestational age effect or influence because we collected samples starting at 12 weeks gestation. So hopefully between that and the timing of delivery, we can see possibly a pattern of expression so that we can kind of hone down on what gestational age weeks we would see the most expression and perhaps target our sampling at that point.
Dr. Bohimsky?
Irina Bohimsky, Yale University. It was interesting, but basically ELISA did not agree with what you found in the N of 1, your pool. I was wondering whether you would rather do the mass spectrometry on more samples. The reason probably that you weren't able to put these together was that what you found using your top-down approach are likely fragments. So ELISA usually identifies the precursor or has different affinity for different fragments. So your quantitation by mass spectrometry would not agree with ELISA. It's a thing that usually happens. So I was wondering if you plan to actually do more mass spectrometry rather than ELISA.
Thank you for your question. We do. We actually also collected serum samples for each of these subjects that have CVF collections. And our hope is that with serum, there's more reproducible, whereas CVF, there's more variation between sampling. And so we're hoping to perform mass spectrometry on the serum samples as well to hopefully identify some proteins of interest and then confirm with ELISAs.
Good morning. Thank you, Dr. Greedy and Dr. Dalton. And I'd also like to thank the Society for the opportunity to present our research regarding the transplacental transfer of glyburide. Prior to 2000, women with gestational diabetes who failed to achieve glycemic control with diet alone were universally treated with insulin. A benefit of insulin is that it does not cross the placenta to a measurable extent at standard doses. There are multiple disadvantages to insulin, including a complicated dosage schedule, an unfavorable route of administration, and reduced patient compliance. Oral agents, however, are easy to administer and are non-invasive. The use of oral sulfonylureas as an alternative treatment modality for gestational diabetes was initially contraindicated in pregnancy, mostly due to the unknown effects on the developing fetus. Although unconfirmed, reports of neonatal hypoglycemia, macrosomia, and congenital anomalies were thought to be the consequence of in utero transfer. Glyburide, a second-generation sulfonylurea, has a high molecular weight, is extensively bound to albumin, and has a relatively short half-life. Typically, these characteristics limit transplacental transfer, making glyburide an ideal candidate for use in pregnancy. The first study to evaluate the placental transfer of glyburide utilized a single cotyledon human placental perfusion model. With the infusion of a supratherapeutic concentration of glyburide, 1,000 nanograms per ml, glyburide levels on the fetal side of the model averaged 26 nanograms per ml, with a range of 6.5 to 52.1. Reported therapeutic peak levels of glyburide at 2 to 4 hours after dose range from 100 to 300 nanograms per ml. The emergence of glyburide as a viable treatment option for gestational diabetes was the result of a landmark study by Langer et al. In this randomized controlled trial, the degree of glycemic control and studied perinatal outcomes were similar in women taking glyburide versus insulin. Using methodology with a limit of detection of 10 nanograms per ml, glyburide was not detected in the cord serum of any infant. And maternal glyburide levels ranged from 50 to 150 nanograms per ml. The pharmacokinetics of glyburide in the third trimester was recently studied in women with gestational diabetes. As opposed to 10 nanograms per ml, liquid chromatography mass spectrometry produced a limit of detection of 0.25 nanograms per ml. Maternal glyburide levels at birth ranged from undetectable to 32.7 nanograms per ml, while umbilical levels ranged from undetectable to 12.5 nanograms per ml, with the authors stating that umbilical levels averaged 70% of maternal levels. The goal of the present study was to determine the magnitude of in vivo transfer of glyburide in women with gestational diabetes. Patients who received care through our Diabetes and Pregnancy program and were treated with glyburide for the management of gestational diabetes were screened for enrollment. Upon admission for delivery, the patient's glyburide dose and time of last dose was recorded. Patients were maintained on their individual glyburide regimen throughout labor or prior to a scheduled cesarean section. Immediately following delivery, a maternal venous blood sample and an umbilical venous blood sample were obtained. Maternal and umbilical cord blood samples were immediately centrifuged for 20 minutes, and the serum was frozen for future evaluation. For patient sample preparation, the samples were thawed, and 100 microliters of serum was vortexed with 20 microliters of the internal standard, 1 nanogram per microliter of glipizide.
400 microliters of chloroform was then added, and the samples were vortexed again, followed by centrifugation at 4 degrees Celsius. After evaporation of the solvent via nitrous gas, the sample was reconstituted in mobile phase. The samples were analyzed with ultra-high performance liquid chromatography mass spectrometry under positive ion mode, and the transitions monitored were 494.1 to 169.1 for glyburide and 446.1 to 321.1 for glipizide. Calibration and analytical standard curves were constructed using the peak area of glyburide and glipizide, and test samples were then interpolated from the calibration curve. The assay was found to be linear from 0.5 to 100 nanograms per ml of glyburide, with a limit of detection of 0.5 nanograms per ml. Maternal and umbilical values were compared by a paired t-test, and correlation was studied by linear regression. 19 sample sets were analyzed. The maternal glyburide dose ranged from 1.25 to 20 milligrams per day, with 53% of patients being on 2.5 milligrams a day. 68% of patients were on a once-a-day dosage schedule. The mean and median glyburide dose was 6.6 milligrams per day and 2.5 milligrams per day, respectively, while both the mean and median time between last dose and sampling was 13 hours. This chart demonstrates the glyburide levels in maternal and umbilical samples. Maternal glyburide concentrations ranged from 0.93 to 70.7 nanograms per ml, with a mean of 15.4 and a median of 6.2 nanograms per ml. Umbilical glyburide concentrations ranged from 0.68 to 32.4 nanograms per ml, with a mean of 7.5 and a median of 3.7 nanograms per ml. The mean ratio of umbilical glyburide to maternal glyburide was 0.83. However, in 15 of the 19 umbilical samples, the concentration of glyburide was less than 10 nanograms per ml. In 7 of the 19 umbilical samples, we had a higher concentration of glyburide than in the maternal sample, but only one of these was greater than 10 nanograms per ml, measuring 10.1. This graph demonstrates the relationship between the maternal glyburide level and the umbilical glyburide levels. And as you can see, as the maternal glyburide level increased, so did the umbilical glyburide level. However, in both the maternal and umbilical glyburide levels, we saw concentrations in the majority of these patients at less than 10. This graph demonstrates the relationship between the total maternal glyburide dose and the maternal glyburide concentrations. And as expected, as the total glyburide dose increased, so did the maternal glyburide concentration. But of note was that at the extreme ends of the total glyburide dose, we noticed high variability in our maternal glyburide levels. This graph represents the relationship between the total maternal glyburide dose and the umbilical glyburide concentrations. Once again, as the total glyburide dose increased, we saw an increase in umbilical glyburide concentrations, but also once again, at the extremes, we saw a high variability in the umbilical glyburide concentrations. From our data, we conclude that transplacental transfer of glyburide does occur. However, umbilical concentrations of glyburide are generally less than 10 nanograms per ml. Recent developments have showed that glyburide is actively effluxed from the fetal compartment to the maternal compartment through the placenta. The breast cancer resistance protein has been implicated as one of the possible mechanisms of this process. Furthermore, there is literature to support that polymorphisms in the BRCP gene may alter the placental pharmacokinetics of glyburide. And while we did note a relationship between maternal and umbilical glyburide concentration levels, the variability in our samples was not expected. We speculate that genetic variation in our patient population and the potential alterations in the function of the BRCP gene may have played a role in the wide variability of our results. I would like to thank everyone who collaborated on this research, including those at St. Luke's Roosevelt Hospital in New York City and those through the Mother Risk Program at the Hospital for Sick Children in Toronto. And a very special thank you to the patients who participated in this study. Thank you.
Paper is now open for questions. Dr.
Hi,
Bakshi.
Bakshi, New York City. Fascinating work. The question which I have is, you measured umbilical vein blood sample, but the umbilical artery is more representative of the fetus rather than umbilical vein. And since you had this outlier in which the fetal level or the umbilical vein level was higher, do you have any explanation besides the genetic?
So,
how would that happen?
I did attempt to measure arterial samples as well. It was just technically very challenging to get enough sample to get an appropriate amount of serum to run. And the samples that we did run, we actually did see similar results in both the vein and the artery. We didn't analyze that statistically. As far as having a higher level in the fetus than in the mother, this was actually also reported in the Hebert study, so it's not completely unforeseen. I don't have a great explanation for that, other than the potential role that these transport proteins may play at the level of the placenta.
Thank you.
Dr. Kustan.
I'm Kustan from Providence. First of all, thank you for doing this study. I think these kinds of studies are really critical as we try to decide whether to use medications in pregnant moms. And it's confirmatory of the Hebert study. And the question I have is that similar to the Hebert study, the patients really didn't have therapeutic the mothers didn't have therapeutic levels of glyburide, at least by the levels you talked about in your introduction. And yet the fetal levels did parallel the mother levels. And the question is, what are the therapeutic or toxic levels of glyburide for a fetus?
We don't know that information. What we do know is that there's some speculation that approximately serum concentration between 30 and 50 nanograms per ml is required to lower blood glucose.
In an adult?
In an adult. Fetal data at this point, we don't have that information.
Programming the pancreas or anything else?
None that I'm aware of.
Thank you.
Yes, sir.
Larry Newman, Kaiser, Oakland. Were there any glucose values on the maternal or fetal set?
Yes, there were. We did gather some neonatal data. And as you can see here, the mean neonatal blood glucose at delivery was 63, with a mean time from delivery to sampling at 29. We also evaluated this comparing the relationship between the umbilical glyburide levels and neonatal glucose, and we found a weak correlation between these two. We didn't define hypoglycemia, but we did not have any infant require admission to the NICU or need for IV glucose infusion.
Dr. Lamb.
University of Tennessee, Chattanooga. I'm curious about the data that you showed looking at umbilical glyburide levels versus total glyburide dosing and maternal serum levels, because the display seems to suggest that we might be missing the sweet spot in terms of overall dosing. The majority of your dosing, about two-thirds, were about 2.5 milligrams per day, yet we see such display in terms of transmission through the fetus that I wonder, should we be actually really looking more at a more standardized higher dose, such as 5 milligrams? And in relation to this, do you have any data that compare neonatal outcomes in terms of macrosomia as well as neonatal hypoglycemia to kind of back that up?
We had no macrosomia in our sample size, in our sample set, and our average birth weight percentile was around 44%. Can you repeat the first question?
I'm sorry. So building on Dr. Kustan's idea, when you look at the display from dosing at the extremes, so at the lower dosing, at the higher dosing, there's a wide variation between what you see in the umbilical blood serum. So what I'm wondering is that if in the middle, indeed, you're seeing more adherence to that linear regression, are we perhaps missing the sweet spot for dosing of glyburide? And for example, instead of just going 2.5 or higher dosing at 20, should we be looking more at a more usual regular dosing at 5 or 7.5 or so forth to see if there's greater compliance, I guess, between the fetal serum levels and maternal levels?
I mean, I think the maternal dosage is going to be based on your blood glucose, what you're getting with your maternal blood glucose. You know, we would like to see minimal amounts on the umbilical side of this model. So I think your maternal dosing is going to be based on your clinical presentation of your glucose values throughout the pregnancy.
Okay.
Thank you, Dr. Schwartz.
Titled The Effect of Maternal Pravastatin Therapy on Adverse Neurologic Outcomes in the Offspring of a Murine Model of Preeclampsia will be presented by Dr. Carver on behalf of her colleagues at the University of Texas Medical Branch in Galveston.
Thank you, Dr. Dalton, Dr. Grady, members and guests. Good morning. I would like to thank the program committee for selecting our scientific work for presentation. In order to study preeclampsia, we have turned to an animal model. We have previously reported on the use of a recombinant adenovirus vector that leads after a single injection in the tail vein to hepatocyte transduction, which allows sustained in vivo expression of the anti-angiogenic SFLT1. SFLT1 is then secreted into the systemic circulation, as can be seen in this graph that represents serum SFLT1 concentrations in red. This leads to maternal hypertension, also in red, and other findings consistent with a preeclampsia-like picture. Barker and others have proposed the developmental origin of adult disease theory, in which not only do maternal and genetic factors contribute to adult diseases, the uterine environment also plays a significant role. We have previously shown, using our animal model, that preeclampsia, by altering the intrauterine environment, leads to long-term changes in the offspring that affect blood pressure, as shown in red in the graph to the right, as well as pup susceptibility to hypoxic ischemic injury in the brain. Additionally, we have shown that pups exposed to preeclampsia have altered expression of markers of neuronal migration, apoptosis, and hypoxia in the brain. Findings in our animal model are consistent with epidemiologic studies showing that children of preeclamptic mothers are at increased risk of abnormal motor and verbal development, ADHD, autism spectrum disorders, and other findings of cognitive delay and decreased academic achievement, particularly when associated with fetal growth restriction. Attempts for primary and secondary prevention of preeclampsia have had limited success. Because of the association and similarities between preeclampsia and adult cardiovascular disease, we and others have focused on the role of pravastatin in preeclampsia prevention. Using animal models, we and others have shown that pravastatin treatment in mice destined to develop preeclampsia leads to reduction in SFLT levels, increase in placental growth factor levels, lowering in blood pressure, improvement of the vascular phenotype, and amelioration of other manifestations of preeclampsia. We also demonstrated that antenatal pravastatin treatment reverses the changes in expression of markers of neuronal migration, apoptosis, and hypoxia in pup brains. These findings have been observed without any adverse effect on pup resorption, deformation, or changes in maternal cholesterol levels. Our objectives were therefore to evaluate long-term neurologic outcomes in adult offspring of our SFLT1 preeclampsia model and to determine whether prenatal pravastatin therapy prevents adverse fetal programming. Timed pregnant CD1 mice at day 8 of gestation were randomly allocated to injection via the tail vein with an adenovirus carrying SFLT1 or adenovirus carrying the murine immunoglobulin FC fragment as our virus control. On the day after injection, the SFLT1 mice were randomly allocated to receive pravastatin dissolved in drinking water or water only. Control pregnant animals received water only. This resulted in 3 groups: virus control mice, MFC, shown in yellow, mice injected with SFLT1 and given only water, shown in red, and mice injected with SFLT1 and treated with pravastatin, shown in blue. For the rest of this presentation, yellow will be used to represent the control group, red the preeclampsia group, and blue the preeclampsia group treated with pravastatin. Mice were allowed to deliver, and treatment assignment was continued until weaning. Offspring were then culled into sibling groups and allowed to grow to adulthood. At 3 and 6 months of age, these adult offspring underwent standardized motor and coordination assays. The negative geotaxis assay was used to evaluate vestibular function and locomotor coordination. Mice were placed in neutral position on a 45-degree plane with head facing downward, and performance was evaluated by time to change orientation by 180 degrees. Mean time to right was used for comparison, and trials were performed 3 times on 2 consecutive days. Balance and fine motor coordination were evaluated using the balance beam test. A beam was elevated above a padded surface, and mice were placed on the beam for 3 trials over 3 consecutive days, and performance was evaluated by number of paw slips and falls. Yes. Balance and motor coordination were also evaluating on a climbing assay. Mice were placed facing upward at the bottom of a 90-degree angle grid and were evaluated over 5 trials on 3 consecutive testing days. Performance was measured by latency to begin climbing, as well as time to reach a top platform, and mean times were used for comparison. Finally, the innate response, which restores our bodies to normal orientation, the righting reflex, was used to evaluate vestibular function. Mice were suspended above a padded surface, and ability to land when released on all feet was recorded. For our analysis, ANOVA with post-hoc comparisons was performed, and ANCOVA was also used to adjust for offspring weight in each assay. A P of less than 0.05 was used for statistical significance. On each graph, time will be reflected on the y-axis, and this is mean time over trials, and on the x-axis, you'll see group by gender. On the negative geotaxis evaluation, which tests vestibular function and coordination, there were no differences between groups or gender at 3 months. However, at 6 months, male offspring born to preeclamptic mothers showed increased time to right compared to the MFC control group. Pravastatin treatment of SFLT mice restored time to right to levels similar to the control group offspring. No difference was seen among the female offspring. On the 3-month balance test, which tests balance and fine motor coordination, female offspring born to preeclamptic mothers had a 4-fold increase in paw slips compared to MFC and SFLT pravastatin treatment group. No difference was seen among males. There was no difference between the groups at 6 months of age. A trend in males was not significant after controlling for weight. As for falls from the balance beam, at 3 months, female offspring born to preeclamptic mothers had more falls compared to the MFC control group and SFLT pravastatin group. This was not significant in males after controlling for weight. There were no differences between the groups at 6 months of age. On the climbing assay, which also tests motor coordination and balance, 3-month-old offspring showed similar times to begin climbing. However, at 6 months, time to begin climbing in female SFLT offspring was significantly longer in comparison to the MFC control group, and pravastatin reduced that time to levels similar to control. A similar trend was seen in males but did not reach significance. As for time to reach the top platform in the climbing test, 3-month-old male SFLT offspring showed significantly increased times compared to the MFC group, and pravastatin treatment again reduced that time to levels similar to control. Female offspring showed no differences in time. There was no difference between the groups at 6 months of age at time to reach the top platform. As for the righting reflex, which evaluates vestibular function, there were no differences between the groups at either 3 or 6 months of age. We also analyzed whether performance in each of the assays was affected by age, as we performed them at 3 and 6 months in the same mouse population. And after accounting for weight and gender, we found that only offspring born to preeclamptic mothers performed worse on the negative geotaxis assay and took longer to begin the climbing assay at 6 months compared with 3 months of age. And pravastatin prevented this worsening with age. In summary, at 3 months of age, male offspring born to preeclamptic mothers performed worse than controls when completing the climbing test. and female offspring performed worse on both parts of the balance test. Whereas at 6 months, male SFLT offspring performed worse in the negative geotaxis assay, and females were worse on the Preeclam assay. Maternal treatment with pravastatin prevented each of these abnormalities in the offspring and restored their performance to levels similar to control mice. So in conclusion, preeclampsia adversely affects motor and coordination function in adult murine offspring in our animal model, and pravastatin prevents this altered developmental programming. I would like to acknowledge my collaborators for their help and support. Thank you very much.
Dr. Carver's paper is now open for presentation, and I would just like to start by asking you, I think this is extremely important work because it represents the potential of a treatment, and if you would just comment on the potential for teratogenicity.
Thank you very much, Dr. Dalton. So that's actually a common question. So statins, as we know, were initially classified as Class X by the FDA when they came onto the market. And by definition, this means that the risk of taking the drug in pregnant women clearly outweighs the benefit. This was based on the fact that previously there were no clear indications of perceived benefit to taking statins in pregnancy, specifically because atherosclerosis is a long-standing disease. It was not felt that discontinuation for 9 months would significantly impact the course of that disease. However, in addition to this meta-analysis looking at all statins, which did not support teratogenesis, another study from the genetics branch of the NIH that reviewed pregnant women exposed to statins showed no teratogenic outcomes associated with pravastatin use specifically. All of the normal outcomes in neonates, pravastatin fell into the category of all normal outcomes in neonates, and there were no anomalies, growth restriction, or intrauterine fetal demise cases associated with pravastatin use in pregnancy. Also, in a prospective observational study that compared women exposed to statins during the first trimester to those not exposed to teratogen, there were no difference in the rates of congenital malformations or other events, and no malformations were associated with pravastatin. In data from a Toronto-based population registry looking at women exposed to statin use in the first trimester to those exposed prior to conception, again, there were no difference in malformations. And I'll just go quickly to data also presented earlier in this week by our colleagues in Galveston looking at a transfer of pravastatin across the placenta. Using a dual perfusion model of the placental lobule, we have been able to see that there is bidirectional transport of pravastatin across the placenta, but the net clearance is actually in the fetal to maternal direction, which is also reassuring. And that pravastatin is subject to efflux transport, which may explain this. All this has actually allowed our site, as well as other collaborators, to obtain FDA approval to begin phase 1 human studies. So we eagerly await those results.
I think you've answered my question. Thank you. Thank you so much.
Thank you.
Spectacular job. Question?
Metro Health in Cleveland. Whenever you put an adult on pravastatin, one of the things you have to do is monitor them for liver dysfunction. You're obviously giving a very high dose of pravastatin to these mice. Has, in this study or in this model previously, any studies been done to assess if pravastatin affects the efficacy of your gene transfer in the hepatocyte vector? Thank you.
We have not specifically looked at liver function testing or gene expression related to hepatocyte transfer. I will say, though, that of the statin family, pravastatin is most selective for hepatocyte uptake and is the weakest inhibitor of the HMG-CoA reductase enzyme. So we feel that of the family, it's probably the safest to use, but we have yet to look at those safety features in our animal model.
Dr. Elowitz?
Thank you. Mikhail Elowitz from Penn. I just want to commend you on using an excellent and extensive animal model and outcome data. I guess I just have one conceptual concern about the model and then a question about your neurodevelopment outcome. So the model, as you have and others have shown with your SFLT, they're actually hypertensive from E12 on. So it just seems like it might be more a model of severe chronic hypertension than actually preeclampsia. So maybe you can speak a little bit to that. And then I guess with the neurodevelopment behavioral outcomes, I guess I'm not as familiar with preeclampsia really causing those kind of neurobehavioral abnormalities in the long term, except if they're preterm. But if they are, and you speak about ADHD and autism, the neurocognitive tests that you did don't actually accurately reflect those neurobehavioral disorders. So I'm wondering what made you pick motor and coordination function for those.
So I'll go to your second question first. So the data that we use to, from a human epidemiologic perspective, actually, to support this animal model was actually reflected in a few recent publications. I'll just mention one in the sake of time. In 2011, in the Gray Journal, a group from the Netherlands actually published work from a prospective cohort study looking at women expectantly managed with severe preeclampsia, HELP, IUGR associated with hypertension between the weeks of 24 and 34. And actually did a battery of tests on 4.5-year-olds. And this included IQ, gross motor function, neurologic testing, and behavioral assays. And when taking out perinatal mortality, there was a 20% composite abnormal outcome on these assays.
Preterm infants. Under 37 weeks.
These were preterm, yes. However, in other prospective cohort studies, when controlling for gestational age at weight and delivery, there's another, they actually still found significant findings with offspring of preeclamptic pregnancies. And can you repeat your first question? I'm sorry.
I think we're going to have to move
on.
on.
Thank you for your time.
Clearly a lot of interest in this topic. And
good morning. We would like to thank the Society for allowing us to present our research. As this audience is well aware, preterm birth remains the leading cause of neonatal morbidity and mortality. Despite research efforts, the rate of preterm birth in the United States has continued to rise over the last 25 years. While the precise pathways that govern preterm parturition remain to be discovered, an obligatory step in the process of parturition, either preterm or term, is a change from uterine quiescence to uterine activity. Considering the importance of uterine activity and contractility in the process of parturition, this process has been the focus of therapeutic strategies to stop preterm labor and prevent preterm birth. Clinical studies with various progestational agents have been performed under the hypothesis that progesterone therapy will promote uterine quiescence. While their mechanisms of action remain unclear, recent studies do support the concept that progesterone supplementation reduces the risk of preterm birth in high-risk populations. 17-alpha-hydroxyprogesterone caproate, or 17P, has been shown to prevent recurrent preterm birth in patients with a prior spontaneous preterm birth. In addition, 2 randomized controlled trials have shown vaginal progesterone reduces the rate of preterm birth in women with a sonographically short cervix. While these clinical studies suggest that progestational agents are a potential therapy to reduce the burden of preterm birth, the molecular mechanisms by which these agents prevent preterm birth remain poorly understood. This lack of understanding how progestational agents prevent preterm birth will limit our ability to have a greater impact on decreasing the preterm birth rate. Throughout pregnancy, the uterus remains relatively quiescent. Myometrial activity is inhibited during pregnancy by a number of substances, including progesterone. During myometrial activation, either preterm or term, there is an increase in expression of contraction-associated proteins, ultimately leading to an increase in uterine contractility and parturition. While contraction-associated proteins have long been associated with uterine activity, a recently discovered family of microRNAs, the miR-200 family, has also been found to be involved in uterine activity. This family of microRNAs has been demonstrated to be induced in the uterus at term in both mice and humans. The miR-200 family is progesterone-mediated and is a regulator of myometrial activity. The targets for the miR-200 family are listed on this slide. How these pathways are integrated and involved in myometrial activity is represented schematically on this slide. At term, the functional decline in progesterone leads to a decrease in expression of ZEB1 and ZEB2. The decrease in ZEB1 and ZEB2 increases the production of contraction-associated proteins leading to labor. The decrease in ZEB1 increases the expression of miR-200A and miR-429. miR-200A targets the transcription factor STAT5B, which serves as a progesterone-responsive transcription repressor of 20-alpha-hydroxyprogesterone dehydrogenase. 20-alpha-hydroxyprogesterone dehydrogenase metabolizes progesterone to an inactive metabolite, 20-alpha-hydroxyprogesterone, further decreasing progesterone in the myometrium. Therefore, Based on clinical studies demonstrating progestational agents can prevent preterm birth in high-risk populations, we hypothesize these agents are modifying pathways that are critically involved in uterine activation. These studies sought to assess if supplementation with progestational agents modified crucial aspects of these pathways as potential mechanisms by which they prevent preterm birth in clinical trials. To test this hypothesis, we used CD1 pregnant mice, which were allocated to 4 treatment groups: 1 milligram of progesterone vaginally, 0.1 cc's of Replens vaginally as a control to vaginal progesterone, 2.5 milligrams of 17p subcutaneously, or 0.1 cc's of castor oil subcutaneously as a control to 17p, with 4 animals per treatment group. Treatments were administered daily on embryonic days 14, 15, 16, and 17. A murine gestation lasts 19 to 21 days, and treatment on embryonic days 14 through 17 mimics late second and third trimester supplementation in humans. On embryonic day 17.5, 6 hours after the final treatment, dams were euthanized. Uterine tissue was harvested, and whole blood was collected. Uterine tissue was collected from all 4 treatment groups. qPCR was performed to assess differential gene expression in the following pathways: the contraction-associated proteins, progesterone-mediated regulators of uterine quiescence, and the microRNAs involved in uterine quiescence and contractility. Progesterone levels were assessed with an enzyme-linked immunosorbent assay kit. We chose to test serum progesterone levels to determine if the administration of either vaginal progesterone or 17p had a measurable increase on circulating levels of progesterone, indicating an increase in bioavailability at the uterus. These are our results. The bar graph shown here depicts the mean and standard error of the mean of messenger RNA expression for the different targets assessed. For each of our targets, 2 graphs will be displayed. The graph on the left shows results for vaginal progesterone, and the one on the right shows the results of 17p. As noted here, mRNA expression of connexin 43, a prototypical contraction-associated protein, was not significantly altered by vaginal progesterone on your left or 17p on your right. Similarly, there was also no difference in expression of the oxytocin receptor with either vaginal progesterone or 17p. Additionally, there was also no difference in the expression of cyclooxygenase 2 with either vaginal progesterone or 17p. This table demonstrates the mean value of quantitative real-time PCR results on the effect of vaginal progesterone on the progesterone-mediated regulators of myometrial contractility. None of the targets assessed were altered with vaginal progesterone. Similarly, none of these studied pathways were significantly affected by 17p. The bar graphs shown here depict the effect of vaginal progesterone and 17p on serum progesterone levels. Both vaginal progesterone and 17p significantly increased serum progesterone levels on embryonic day 17.5 when compared to Replens and castor oil, respectively. Treatment with either agent raised progesterone levels 3 to 4-fold compared to previously published murine progesterone E17 levels. A notable strength of our study is that we explored the effect of vaginal progesterone and 17p on multiple uterine pathways regulating contractility and quiescence as a possible mechanism by which progestational agents prevent preterm birth in the clinical realm. Our results indicate treatment with these 2 agents did in fact raise circulating progesterone levels in our mouse model. Despite the increase in progesterone levels, these agents had no notable effect on gene pathways involved in uterine activity and quiescence. The animals in this study received a 25-fold greater dose of vaginal progesterone and a 17-fold greater dose of 17p compared to human dosing. Despite these superhuman doses, neither vaginal progesterone nor 17p had any measurable effect. Our study has some limitations. While we explored many pathways known to be involved in uterine contractility, there are likely other pathways, known and yet to be discovered, involved in the process by which the uterus changes from a quiescent to an active state. However, our exploration was fairly comprehensive and investigated those pathways reported to be of significance based on available evidence regarding uterine contractility in term and preterm labor. Another limitation is that we did not investigate changes in protein levels. While alterations in protein levels can occur without changes in transcription, this is not a common process. Furthermore, in other in vivo models and in vitro studies, treatment with exogenous progesterone has been demonstrated to alter mRNA expression as rapidly as 30 minutes and undoubtedly by 6 hours. As we gave high and repeated doses of progesterone, we would expect that progesterone would be able to alter gene pathways in the time period that we studied. As such, with no changes in mRNA expression, we did not pursue additional protein studies. This work focused on the effect of vaginal progesterone and 17P in a normal pregnancy. We did not study the effect of these agents in the setting of an inflammation-induced preterm birth. If this is the mechanism by which these agents prevent preterm birth, these studies were unable to address this possibility. In summary, these studies suggest that neither vaginal progesterone nor 17P had a significant effect on gene pathways involved in uterine activity in a normal murine pregnancy. These studies draw into question the mechanisms by which these agents prevent preterm birth in high-risk women. Our results may explain why progesterone has failed to alter the preterm birth rate in twins or in women with moderately short cervices, but not very short, as these women may be at risk for preterm birth from activation of non-inflammatory pathways. Although both vaginal progesterone and 17P are used to prevent preterm birth in high-risk patients, the mechanism of action of these agents has yet to be uncovered. Further studies are needed to determine the molecular mechanisms by which these agents prevent preterm birth. By continuing to increase our understanding of uterine pathways regulating contractility and quiescence, we may be able to identify novel therapies to prevent preterm birth. I would like to thank Monique Maubert and Dr. Anton, who taught me many of the techniques needed to perform much of this work. I'd also like to thank Dr. Elvitz for her mentorship and guidance. Lastly, I would like to thank the Maternal and Child Health Research Fund at the University of Pennsylvania for funding this work. I'd be happy to answer any questions at this time. Thank you.
Thank you, Dr. Knoll. This paper is now open for questions. Yes, sir.
Dinesh Shah, University of Wisconsin, Madison. I'm curious if any of the genes that you interrogated, are they directly regulated by progesterone, as in, is there a progesterone response element upstream in the DNA of these genes, or are they indirectly regulated, and do you know how they're regulated as far as progesterone is concerned?
Well, the progesterone-mediated regulators of uterine quiescence and the microRNAs appear to be indirectly regulated. The STAT5, I'm going to go back for a second here. So the ZEB1 and ZEB2 appears to be directly regulated, and these ZEB1 seems to affect the production of contractile-associated proteins. But further downstream, you can see all these pathways seem to be indirectly regulated.
Do you know they have the response elements?
Do I know, do they have response elements? Yeah, prior studies have shown that ZEB1 and ZEB2 is regulated, is progesterone-mediated, and then the pathway follows suit from the progesterone supplementation.
I have a very naive question. Is it possible that the mechanisms by which progesterone may work are not activated until the inciting event leads to the onset of labor?
It's very true. Thank you for the question. In this study, we looked at a normal mouse pregnancy. So we were just looking at the effect on these pathways in a normal pregnancy. So if these pathways are activated prematurely from an inflammatory process, I can't answer that question based on what we've done here. But I have plans to order the mice and do that study. So.
Okay. Any other questions? Thank you very much.
Daniel Krause on behalf of his colleagues at Duke.
Good morning. Thank you for the opportunity to present this research. This project started with a simple clinical question. How does smoking protect against preeclampsia? Preeclampsia is thought to be due to poor trophoblast invasion. The 3 panels here illustrate this process. During normal placentation, on the right, trophoblast cells invade the muscular layer of the spiral arteries, leading to vasodilation and increased perfusion in the placental bed. This process requires that cells invade through a basement membrane composed of collagen and other structural elements. The middle panel here illustrates preeclampsia, characterized by poor trophoblast invasion, leading to poor perfusion in the placental bed and the release of placental factors that trigger endothelial cell dysfunction. Epidemiologic data indicate that smoking significantly reduces one's risk of preeclampsia. This effect is quite strong, with a meta-analysis demonstrating an odds ratio of a smoker developing preeclampsia of 0.51. Frustratingly, we don't fully understand how smoking reduces preeclampsia risk. We do know that in order for smoking to be protective, exposure must continue through the third trimester. And furthermore, the tobacco must be smoked, because non-combustible forms of tobacco use, such as snuff or chew, don't offer protection. Prior work in our lab has linked cigarette smoke's effect on trophoblast cells to the hormone adrenomedulin. Adrenomedulin is a peptide hormone that has many biologic effects, including being vasodilatory and proangiogenic. It's expressed in the reproductive tract, placenta, and fetal membranes, and has been shown to have altered levels in preeclamptics compared to normal controls. These images show representative casts of the vasculature of mouse placentas. The adrenomedulin knockout placenta on the right demonstrates decreased branching angiogenesis compared to the wild-type mouse placenta on the left. Furthermore, the decidua of mice lacking adrenomedulin exhibit decreased number of natural killer cells. Both of these findings are common human placental findings in preeclampsia. Exploratory work in our lab treating trophoblast cells with cigarette smoke extract demonstrated a significant upregulation in the gene transcripts of the vasoactive peptides VEGF and adrenomedulin. Therefore, for this project, we tested two hypotheses. First, we hypothesized that cigarette smoke extract increases the invasiveness of trophoblast cells in vitro via a process mediated by adrenomedulin, and secondly, that adrenomedulin expression is higher in the placental tissue of smokers compared to non-smokers at term. To test the first hypothesis, immortalized HTR8 SB neotrophoblast cells were incubated and allowed to invade through a matrigel invasion chamber for 24 hours in the presence of 6 different treatment arms. Non-stimulated or untreated cells were compared to cells treated with the competitive adrenomedulin inhibitor alone, which is a truncated adrenomedulin molecule that binds to but does not activate the adrenomedulin receptor, or cells treated with adrenomedulin alone, or adrenomedulin co-treated with the adrenomedulin inhibitor, or cells were treated with 1% cigarette smoke extract alone, or cigarette smoke extract co-treated with the adrenomedulin inhibitor. Excluding the bronchial tree, cells in the body of a smoker are exposed to only the soluble components of cigarette smoke. Therefore, cigarette smoke extract is a well-established technique for modeling cigarette smoke exposure in vitro. Cigarette smoke extract was prepared by pulling the smoke of 3 Marlboro Red cigarettes through 50 milliliters of culture media with the aid of a vacuum. The concentration of cigarette smoke extract was normalized to the measured nicotine concentration in order to maintain consistency between batches and across treatments. 1% cigarette smoke extract was found to have 130 nanograms per milliliter of nicotine, which is similar to the blood levels found in a heavy smoker. Matrigel invasion chambers were then used to test for cell invasiveness. A matrigel invasion chamber consists of an insert that's housed in the well of a 24-well plate. The insert has a porous membrane with 8 micrometer pores that is then coated with a layer of matrigel. Matrigel simulates a physiologic basement membrane because it consists of collagen, laminin, connectin, and other connective tissue components. Trophoblast cells were then plated in the invasion chamber in a serum-free media, and then the insert was lowered into the culture well, with the lower chamber containing media with 5% serum, acting as a chemoattractant for invasion. Cells plated in the top chamber were then given 24 hours to invade through the matrigel to the underside of the membrane. Non-invasive cells were then removed from the upper chamber with a sterile cotton-tipped applicator, and cells that had invaded were quantified with an MTS assay. Now, in order to control for altered cell migration with our treatments, cells were similarly incubated in inserts that consisted of only the 8 micrometer porous membrane without matrigel coating, thereby allowing cells to migrate across the membrane without having to also invade through the matrigel. Invasive cells were then stained and imaged with a microscope for descriptive analyses. To quantify our results, percent invasion and an invasion index were calculated for each treatment arm. Percent invasion equals the number of cells invading the matrigel-coated insert divided by the number of cells migrating across the non-matrigel-coated insert for each treatment arm. The invasion index for each treatment is equal to the percent invasion for a treatment group divided by the percent invasion for the unstimulated group. Comparison of invasion indices was performed using a one-way ANOVA, and we performed a Bonferroni correction for multiple comparisons. The results were as follows. These panels show representative images of the cells that successfully invaded the matrigel inserts. Cells are stained blue here, and the small black dots that you see represent the 8 micrometer pores that are present in all of the membranes. The upper left panel shows non-stimulated cells, and the lower left panel shows cells treated with the competitive adrenomedullin inhibitor alone. There's no difference in cell density, indicating that the adrenomedullin inhibitor doesn't alter invasion when given alone. The upper middle panel shows cells treated with adrenomedullin, and the lower middle panel shows cells treated with adrenomedullin and the adrenomedullin inhibitor. There's increased invasion with adrenomedullin treatment alone, and co-treatment with the inhibitor attenuates the increased invasion seen with adrenomedullin treatment alone. The upper right panel shows cells treated with cigarette smoke extract alone, demonstrating increased invasion compared to non-stimulated. And in the lower right panel, co-treatment of cells with cigarette smoke extract and the adrenomedullin inhibitor reversed the increased invasion seen with cigarette smoke extract treatment alone. We next compared invasion indices. Setting the non-stimulated to a reference value of 1, treatment with the adrenomedullin inhibitor alone didn't alter invasion. Adrenomedullin treatment alone increased invasion by 1.5-fold compared to non-stimulated, while co-treatment with adrenomedullin and the adrenomedullin inhibitor significantly reduced the increased invasion seen with adrenomedullin treatment alone. Cigarette smoke extract treatment increased invasion compared to non-stimulated by 1.4-fold, and co-treatment of cells with cigarette smoke extract and the adrenomedullin inhibitor reversed the increased invasion seen with cigarette smoke extract treatment alone. The results from this experiment support two conclusions. Both adrenomedullin and cigarette smoke extract increased trophoblast invasion in vitro, and increases in trophoblast invasion seen with cigarette smoke extract appear to be mediated at least in part by adrenomedullin. Now, given the earlier epidemiologic data that smoking must continue through the 3rd trimester to be protective against preeclampsia, we tested our second hypothesis, that adrenomedullin expression is higher in the placental tissue of smokers compared to non-smokers at term. Using the Duke Pregnancy and Tissue Repository, we identified smokers and non-smokers, all who did not have preeclampsia, who delivered at term, for whom clinical characteristics including smoking status were available. Placental tissue underwent immunohistochemical analysis using a mouse monoclonal anti-adrenomedullin antibody at a 1:100 dilution. 10 fields per subject were imaged with a digital microscope by a blinded researcher. Each high-powered field was then scored for the intensity of adrenomedullin immunoreactivity by 3 independent and blinded reviewers using a semi-quantitative 4-point scale. A mean adrenomedullin intensity score for each subject was calculated, and after proving normalcy for the distribution of subject scores within the 2 groups, a mean adrenomedullin intensity score for each group was calculated and compared with a student's t-test. These summary statistics show that smokers were younger than non-smokers, but that there were otherwise no statistical differences regarding delivery route, gestational age, or newborn birth weight. These upper panels show representative images of the brown staining for adrenomedullin in the placental tissue of a smoker on the left and a non-smoker on the right. The brown staining in the trophoblast cells lining the tertiary villi is more intense in the smoker. When quantified by all 3 independent observers, the mean score was 40% higher for smokers, shown graphically in the lower panel. From this experiment, we concluded that the placental tissue from smokers at term has higher adrenomedullin levels compared to non-smokers. From our work, we demonstrate that cigarette smoke extract treatment of trophoblasts increases cellular production of adrenomedullin, which in turn increases cellular invasion in vitro. Taken together, these experiments shed new light as to how smoking alters trophoblast cell biology. It provides a possible mechanism by which smoking, even in the 3rd trimester alone, is protective against preeclampsia, and suggests that the adrenomedullin pathway is a potential target for the treatment and/or prevention of preeclampsia. Thank you.
Thank you very much. This paper is now open for questions. Oh, Adam, Dr. Urato. We missed you earlier.
Adam Murato from Tufts in Boston. Very interesting work, interesting presentation. It seems, though, that you're almost making an argument for enhanced trophoblast invasion and development of the placenta in smokers, but we know that that's actually not true. For example, if we look at first trimester PAP-A levels, they're decreased in the smokers. And it looks like the early onset preeclampsia, the smoking may not be protective of that, so that there may actually be more of a downstream third trimester effect. For example, we know smokers have smaller babies, less antigen exposure, smaller placentas laid. And it seems to me that this argument almost hinges on smokers having better invasion, healthier placentas, and that's why they're not getting the preeclampsia.
So I think what we tried to do is evaluate two different ideas. One is definitely that first trimester model of increased invasion, but the second experiment was really focused on looking at adrenomedullin levels at term, with the idea that circulating levels may be different, and therefore there may be protection offered just by women who smoke in the third trimester alone, thereby not requiring that first trimester model of increased invasion. Thank you.
Thanks very much.
Excellent question. Final question.
Very nicely done. You may have to go to Washington and Colorado to follow up on.
Thank you.
Thank you very much.